Hi everyone, this is Binyam. Today I'm going to presenting you about vitamin D metabolism. Vitamin D for human being can be obtained from fatty fish. Fatty fish is the main source of vitamin D, and the people who are consuming fatty fish such as sardine, salmon, and some other fatty fishes can have an enough vitamin D. And also there are different kinds of products which are fortified with vitamin D, for example milk, oil, and yogurts and some other products, especially children milk formulas are also fortified with vitamin D. So vitamin D is very important nutrient. So the main source are fatty fish and fortified products. These are the main vitamin D sources. Dietary vitamin D can be obtained in two forms, existed in two forms, either in the form of D2 or D3. In the body, vitamin D do not require any digestions because it's only formed in its free form. It's not bounded with anything. So no digestion is required in the body, both in the mouth, in the gastrointestinal, or in the stomach, or also in the small intestine as well. 50% of vitamin D can be absorbed, and the largest amount of absorptions can take place in the jejunum, in the middle part of the small intestine. And some people may lose this jejunum part due to like weight loss surgeries. So absence of jejunum will lead to vitamin D deficiency. So care has to be taken. So that after the vitamin D is ingested, it will reaching into the duodenum first. In the duodenum, together with other fat molecules. As you may know, vitamin D is a fat-soluble vitamins, so that it will make a micelle, it will form micelles with different kinds of lipid materials, such as triacylglycerol, lysophospholipid, free cholesterols, and also free phytosterols, and also bile acids. These all together will form a micelle. The micelle is used to transport this collective materials such as fat-soluble vitamins, including vitamin D, A, E, K, depending on their availabilities in our diet, lysophospholipids, free cholesterol, and long-chain free fatty acids, medium and short-chain fatty acids, and phytosterol together with the additions of bile acid will form this micelle. This is a representation of micelles. The purpose of micelle is to translocate this group of molecules into the enterocytes apical border or the brush border. In the brush border, vitamin D will diffuse or get into the enterocyte through passive diffusion. And after it's getting to the enterocytes, there is a process called making chylomicron. Chylomicron is formed by different lipid materials such as phospholipid and also triacylglycerol. These two important molecules, after they get into the enterocyte in the form of free fatty acid form and monoacylglycerol form, after they get into the system in the endoplasmic reticulum, there undergoes like kind of reesterification, so that they will produce phospholipid again, triacylglycerol again, and also what was that, cholesterol esters. These things will be reesterified again in the endoplasmic reticulum, and they be used for the formations of chylomicron. Chylomicron is a collection of many nutrients that include triacylglycerol. About 80% of it is triacylglycerol. It also contains cholesterol, phospholipids, and phytosterols as well, and also fat-soluble vitamins, including vitamin D. So this chylomicron is a little bit big, and it's bound by ApoB48 lipoproteins. These lipoproteins help it to transport through the aqueous system, because apolipoproteins have both ends, both hydrophilic and hydrophobic end. So in the hydrophilic side, which is a water-loving side, the chylomicron has the ability to transport it through the lymphatic system. Unlike other nutrients, chylomicron do not directly go into the liver through hepatic portal vein. Rather, they pass through the exocytosis form through the basolateral membrane, and they join into the lymphatic system. In the lymphatics, after they join the lymphatic systems, they will travel to the thoracic vein. It's a vein which is like found in the left sides of the neck, and through this lymphatic system and through this thoracic vein, they will join the blood circulation. After they join the blood circulation, these chylomicrons will be distributed throughout the body and reached into the target cell for further digestions and degradations. Then these chylomicrons will be attached to the non-hepatic cell first, that's found in the muscle or adipose or organ tissues before they're reaching to the liver. But it has to be non-hepatic cell, possibly the adipose tissues. And while they are traveling, the chylomicron will bind to ApoB48 first. That's the original lipoproteins. In addition to that, they have ApoA lipoproteins, but in the blood they will receive ApoC, apolipoprotein C from HDL, and in the non-hepatic cell they have in their periphery they have apolipoprotein C in their cell membrane. So these ApoC apolipoproteins will bind together, so that in the periphery of the cell there are enzymes called lipoprotein lipase, and this lipase enzyme will digest the triacylglycerol that is found in the chylomicron central part, which contains like 80% of it. Then these triacylglycerols will be hydrolyzed by lipoprotein lipase and get into the cell for free fatty acid metabolism. However, the chylomicrons will not get into the cell, but it will be highly reduced. So the remaining part is called chylomicron ruminants, or the remaining parts will be traveled to the liver for further degradation and metabolism. When it comes to the vitamin D will also be existed in these chylomicron ruminants, and this vitamin D3 will be get into the liver cell through the chylomicrons. Chylomicrons have a special attachment places in the liver, which is LDL receptor. The LDL receptors will also accept or receive chylomicron ruminant, and in this way the chylomicrons will get into the liver cell. Then the vitamin D3 in the D3 form will be translocated into the mitochondria, where it undergoes some kind of hydroxylation. Hydroxylation means okay, before I explain this, we have another vitamin D3 sources. So this chylomicron vitamin D3 is a kind of vitamin D3 which is coming from the food we consume, as we start from here. But we have another source of vitamin D, which is sunshine-induced vitamin D. As you may know, in the sun we have ultraviolet B, and when this ultraviolet B is reaching into our skin, we have epidermis and dermis. Then inside between these two important skin components, we have a gland called sebaceous gland. This sebaceous gland secretes 7-dehydrocholesterol hormones, These hormones are very important, and it will be diffused into the epidermis part outside of the skin. And when this 7-dehydrocholesterol and ultraviolet ray B is fused or interact with each other at the specific wavelengths of 285 up to 320 nanometer, they will produce pre-vitamin D3 inside on the skin. And this vitamin D3 will diffuse, get into the skin, under the skin, and from several hours to several days, this pre-vitamin D3 will be converted into vitamin D3. This is how we get another vitamin D3 to our body, and this vitamin D3 will be diffused into the blood circulation system by binding with vitamin D binding protein. This vitamin B binding protein, as you can see, they have like a kind of boat with a green boat. It's nice and comfortable. Then it's traveling through the blood circulations and reaching into the liver again. So we have two sources of vitamin D3 here, from the food and also from the sunshine induced vitamin D3. So both of these D3 will get into the liver mitochondria, where they get hydroxylations. The most important reaction that vitamin D3 in the form of vitamin D3 undergone is that called hydroxylation. Hydroxylation means the additions of OH group to a molecule. So that in the original vitamin D3 form, one OH group is added, and this addition is conducted by the enzyme called hydroxylase enzyme, or 25-hydroxylase enzyme. This is important. 25-hydroxylase enzyme is an enzyme which add one OH group to the 25th carbon atom of vitamin D3. So that when this OH group is added to the vitamin D3 at 25 carbon, we call it 25-hydroxy-OHD, or 25-hydroxy-cholecalciferol. This one is a very important form of vitamin D, which is circulating in our blood, and in most of the time for checking the vitamin D status, 25-hydroxy-OHD is the one we are going to measure. Then this 25-hydroxy-OHD form of vitamin D3 will go out of the liver through the vena cava, reaching to the right arterium, then it's going to the lung for oxygenations, because blood which is carried by the inferior vena cava is the one which is deoxygenated. So that for oxygenation purposes, inferior vena cava you travel or use, transport those nutritious nutrients from the liver to the heart, and the heart will send it to the lung, and in the lung, due to respiration processes, the blood will be oxygenated. Oxygen is very important for nutrient metabolism. Then it will be distributed throughout the tissues. So every tissues in our tissues will receive 25-OHD form of vitamin D3. But the most important site for this 25-OHD or 25-hydroxy-cholecalciferol form is kidney. In the kidney, this form of vitamin D will be attached into its transporter, which is vitamin D binding protein. It will travel to the kidney. In the kidney, there will be a special attachment for vitamin D binding proteins, which is called tubulin megalin receptor. And once these tubulin megalin receptors receive this 25-OHD, the vitamin D in the form of 25-OHD will get into the mitochondria of the kidney. Inside the mitochondria of the kidney, this 25-OHD form of vitamin D will again hydroxylated by another enzyme called 1-hydroxylase enzyme. 1-hydroxylase enzyme. This hydroxylase enzyme is found in the kidney mitochondria, and what this enzyme will do is adding one OH group at carbon number 1. So they call it 1-hydroxylase enzyme because it has the OH group in the first carbon atom. This hydroxylation process will produce a vitamin D with 2 OH group, so that the name will be 1,25-OH2D in short, or 1,25-dihydroxycholecalciferol, or in short, calcisterol or calcisterol. We call it calcitriol. Calcitriol in short. So in the blood circulations, this calcitriol or 1,25-OHD will be transported through the blood circulations by attaching itself with vitamin D binding protein. Calcitriol in the blood have a half-life of from 2 hours up to 6 hours. Calcitriol bind to VDPP to transport tissues. So this is the active form of vitamin D, which is calcitriol or 1,25-dihydroxycholecalciferol or calcitriol. This calcitriol has a lots of target tissues that includes small intestine, bone, kidney, muscles, pancreatic beta cells, brain, skin, hematopoietic system, and central nervous systems are among the most important site. Okay. However, how this calciferol entering into the cell for use is not fully understood. So we don't know how this calciferol is getting into the cell for its action. Vitamin D in the form of calcitriol has a lot of functions, but the main purpose of vitamin D, which is very important, or of course all of them are very important, but the most important one is controlling the serum calcium homeostasis. Serum calcium homeostasis is very, very, very important. So vitamin D is required for this. And the most important actions of vitamin D3 or calciferol is that by improving or increasing the absorptions of calcium in the intestine. After the calcitriol is made by the kidney, it will be circulated throughout the body, including the intestine. And in the intestine, this calcitriol will enhance the synthesis of calcium transporter 1. This calcium transporter 1 is located in the enterocytes brush border, which is located there. For the main purpose of this calcium transporter is to let the calcium ion to get into the enterocytes, so that this transporter synthesis requires calcitriol. The second one is within the enterocytes. It enhances cellular calcium transport from the cytosol to getting out of the basolateral membrane. Here there is a special transporter within the enterocytes, so that special transporter requires calcitriol for its action. The next one will be the extrusions of calcium across the basolateral membrane. So the digested or the absorbed calcium has to be go out through the basolateral membrane to get into the circulation system for its action. So that these extrusions of calcium across the basolateral membranes are enhanced by calcitriol, by enhancing the synthesis of calcium ATPase pump. Calcium ATPase pump is the one which is helping the calcium to be out of the basolateral membrane. In addition to that, calcium undergoes a paracellular absorptions when the concentration is high. So calcitriol or vitamin D3 is controlling this paracellular absorptions of calcium. So by doing these four important actions, vitamin D3 or vitamin D or calcitriol increases the absorptions of calcium, which is very, very important. The second actions of calcitriol is that the mobilizations of calciums out of the bone. This is very important because when we are in short of calcium in our blood, that calcium has to be supplied, or the serum calcium has to be supplied, which is around less than 1% from the total calcium, but a little bits of reduction in calcium in our serum will have a huge consequences that include seizure and daze and muscle cramp. So that the calcium has to be supplied consistently throughout the blood, so that homeostasis is like controlled by almost vitamin D or calcitriol, or the active form of vitamin D3. So by mobilizing the calcium out of the bone, it will help us to maintain our calcium homeostasis. However, vitamin D has a lots of multiple functions that you need to read, Bini. Thanks.